Assalamu alaikum dear students, I am Anjum Shafiq, your biology teacher for grade 10, and today I am going to discuss the exercise of chapter number 17. So let's start. So dear students, first of all we are doing the multiple choice questions. So the first MCQ is find the correct match for the fermentation product and the organism involved. So हमने correct organism और उसका product ढूंढना है. So formic acid Saccharomyces, no dear students, that is not correct. Beta option is ethanol and Saccharomyces. Yes, my dear students, that is the correct option for this question. That ethanol, the ethyl alcohol, is formed from the Saccharomyces cerevisiae. So that is a yeast, a fungus. So ethanol and aspergillus, no, my dear students. And delta option says glycerol and aspergillus. No, dear students, that is also not correct. So the correct option for this question is beta option. It says ethanol and the Saccharomyces. Now we move to another question. Which one is not an objective of genetic engineering? नीचे दिए गए ऑब्जेक्टिव में से कौन सा ऐसा ऑब्जेक्टिव है जो कि जेनेटिक इंजीनियरिंग का नहीं है? So the option delta is correction of genetic defects in higher organisms. Yes, my dear student, that is the basic objective of the genetic engineering. हमसे पूछा गया है कौन सा objective नहीं है. So लेकिन correction of genetic defects in higher organisms through gene therapy, that is the objective of genetic engineering. So that is not our answer. Option 40 says production of RNA and protein molecules. So that is also the objective of genetic engineering, and it is not the required answer of our question. Option beta says isolation of a particular gene or part of a gene means the isolation of gene of interest. So that is also the objective of genetic engineering, and it is also not the answer for our question. We are looking for the objective which is not the objective of genetic engineering. So option alpha is production of cheese and yogurt by lactic acid bacteria. So dear students, that is not the objective of genetic engineering. That is the old genetic engineering or old biotechnology which is being used from hundreds of years. So that is not the objective of genetic engineering. So the correct answer for this question is alpha, that is the production of cheese and yogurt by lactic acid bacteria. So we have another MCQ. Which of these is an antiviral protein? So option alpha is urokinase. No, my dear students, the urokinase, that is basically an enzyme that is required or which is used for dissolving the blood clots. Option beta is thymosin. So that is a hormone that is used against the cancer. Option Charlie is insulin. That is also a hormone which is genetically engineered insulin, a hormone which is used in the sugar patients for lowering the blood glucose levels. And the only option remaining is delta, that is interferon. Yes, dear students, interferons are the antiviral proteins which are used against some viral disease like hepatitis. So the correct option for this question is delta, that is interferon. Now let's move to another question. The first step in genetic engineering is. So dear students, हमने डिटेल के साथ genetic engineering के steps पढ़े थे. So सबसे पहला step क्या था? Hopefully आपको recall हो गया होगा. The सबसे पहला step था, was it the growth of genetically modified organism? No. Was it the transfer of recombinant DNA into the host organism? No, my dear students. Or the isolation of gene of interest? Yes, dear students, that was the first basic step in genetic engineering, that is the isolation of gene of interest by using some enzymes which are called as endonucleases or restriction endonucleases, which are used to cut the DNA at a specific site. And delta option is insertion of a gene into vectors, so that was the second step. So the first step is Charlie, that is isolation of gene of interest. That is the answer of our question. Here are some short questions. Define biotechnology and describe its importance. So dear students, biotechnology is defined as the use of living organisms in the processes for the manufacture of useful products or for services. Biotechnology play important role in our daily lives, in the field of medicine, in the field of food, agriculture, and in the betterment of our environment. Here is another question. What is a fermenter? What are the two types of fermentation carried out in fermenters? So dear students, the fermenter is a device that provides an optimum temperature or optimum pH, or we can say the optimum environment for the microorganisms to grow into biomass so that they can interact with the substrate, forming our desired product. And the fermentation is carried out in fermenters in the following two ways. The first one is batch fermentation, or which is also called as discontinuous fermentation, and the second type is continuous fermentation, which we have discussed in the lectures in detail. Here is another question. Describe the achievements of genetic engineering in medicine, agriculture, and environment. So various achievements of genetic engineering in medicine, agriculture, and environment are as follows, like human insulin, that is a hormone which is genetically engineered to be used by the diabetic patients. Human growth hormone or somatotrophic hormone that is used to increase the height of a human being. Thymosin, that is a hormone which is genetically engineered, and it is produced by the brain cells against the lung cancer and the brain cancer. So thymosin, it is a hormone which is used for the treatment of cancers. Beta endorphin, it is a painkiller which is naturally produced by our brain cells, but now it is genetically engineered to be used in cases of acute pain. Vaccines, dear students, we have discussed it in detail, the materials which contain the dead or weakened microorganisms which boost up or which provoke our immune system to make the antibodies against them or to mobilize the cells. Genetically modified animals and plants, these plants fix nitrogen from atmosphere and required fewer fertilizers. Now we have another question. It says, what basic steps a genetic engineer adopts during the manipulation of genes? The basic steps of genetic engineering are: number 1, isolation of gene of interest. Number 2, insertion of gene of interest into a vector. Number 3, the transfer of recombinant DNA into the host organism. Number 4, growth of the genetically modified organism or GMO. And finally, the 5th step is the expression of the gene. So here we have another question. What are single cell proteins? Describe their importance. So that is the last topic of our chapter, in which we have discussed the single cell proteins. The proteins produced by the unicellular microorganisms is called the single cell proteins or novel proteins. How would you define fermentation with reference to biotechnology? So fermentation is defined as a chemical change brought about using microorganisms. For example, in the biotechnology industry, for the production of pharmaceuticals, food additives, and animal feed stuffs. Here is another question. Name any 2 industrial products made by fermentation. Also describe their uses in the industry. So we have discussed 4 of them. Here we are discussing only 2. The first one is formic acid. It is used in the textile dyeing, leather treatment, electroplating, rubber manufacture. And the second one is ethanol or ethyl alcohol. It is used as solvent, organic solvent, used in the production of vinegar and beverages. Here we have another question. What are the products of 2 types of carbohydrate fermentation? So while there are a number of products from carbohydrate fermentation, the most common are ethanol or ethyl alcohol, and lactic acid. Here we have another question. What are the products of two types of carbohydrate fermentation? Biotechnology is also being used for dealing with environmental issues. Bacterial enzymes are used to treat sewage water to purify. Microbes are being developed to be used as biopesticides, biofertilizers, biosensors, etc. We have discussed it in detail in our lectures. Such transgenic microorganisms are also used for the recovery of metals, the cleaning of spilled oils in the oceans or in the rivers, and for many other purposes. Here is another question. In biotechnology, what is meant by genetically modified organisms or GMO? How is it made? So a genetically modified organism or GMO is an animal, a plant, or a microbe whose DNA has been altered. Altered means changed using genetic engineering techniques. It's called as a GMO. So dear students, hopefully आज का लेक्चर भी आपको क्लैरिफाई हो गया होगा. वो तमाम कन्फ्यूजन्स जो कॉन्सेप्ट वाइज थी वो भी क्लैरिफाई हो गई होंगी. सो मिलते हैं नेक्स्ट लेक्चर में. तब तक अपना ख्याल रखिएगा. अल्लाह हाफिज़.